Hello everyone, today we'll be talking about the sequentially bidirectional gastrovascular flows in highly branched digestive tracts of planocerid flatworms. In our study, we experimented with the polyclad flatworm planocerid oligoglenna. These are some of the objectives we aim to tackle, including the confirmation of its predation stages, the observation of its gastrovascular flow dynamics, and the assessment of the function of its small muscular valves in the bifurcated branches. In order to observe the behaviors of flatworms, we videotaped them for long periods of time, and to further examine their digestive tracts, we also developed a staining technique adopted from that of whales in 2017, which we used on clams that we then feed to flatworms, thus coloring their branches. To observe these stain tracts, we had static image processed to mark the blue regions. Post-stain active tracking was then used to analyze the stain ratio within a selected section of the tracts. Through long-term video recording, we discovered that Planocerid's predatory behavior can be divided into three periods: attack, invasion, and ingestion. Next, focusing on the movements of the digestive tract during the ingestion period, secondary staining and static image analyses reveals the complex structures of the tracts and suggests a higher order of branches than visible to the naked eye. Post-stain active tracking further reveals sequentially bidirectional gastrovascular flows for the first time. We also observed bidirectional digestive movements parallel to that of the hydra as observed by Shimizu in 2004, including segmented movement and peristalsis. Here we tracked 4 orders of the branches in both directions. It can be deduced that the trough of the curve represents the duration of contraction. This result also suggests that flatworms do not rely solely on pharynx contraction for transporting food. Fixed segment positions contract regularly and sequentially, resulting in rhythmic movement patterns in both directions. Regardless of the direction, a movement remains consistent and sequentially bidirectional, albeit at an uneven rate. The digestive movement of the flatworm decreases after about 1 day and stops within 2 days, suggesting these movements primarily serve digestive functions rather than circulatory roles. To understand the anatomical structure, we conducted observations on serial sections of the tract. Our findings reveal that circular membrane-like muscle structures are evenly distributed along the tract. We infer that these circular membranous muscles serve as contracting and pushing forces within the digestive tract. Flatworm static behavior, coupled with the method used in our study, amplifies the visibility of their digestive tracts beyond the naked eye. This approach suggests potential applications for studying various flatworm species. The bidirectional peristalsis, facilitated by circular membrane-like muscles and regulated by a more intricate nerve net compared to hydrozoans, results in constant speed transfer of food. These are our conclusions. And these are some of our references. Thank you for listening.